Hello, today we will be discussing structure of DNA as well as how forces interact to stabilize double-stranded DNA. Watson and Crick discovered the structure of DNA as double-stranded, helical, and containing nucleic acids. Here we have the genetic structure for deoxyribose, one of the parts of a nucleotide. It contains 5 carbons, each labeled 1' through 5'. The 5' carbon connects to a negatively charged phosphate group. This connection between them is what we call a phosphate backbone, which provides structure to the whole molecule. The next part of a nucleotide is the nitrogenous bases, in which there are 2 types, pyrimidines and purines. Pyrimidines consist of cytosine and thymine, and purines consist of adenine and guanine. Base pairing occurs between complementary bases. A pairs with T and C pairs with G. They connect in a similar fashion to how a zipper works. These bases are repeated in different orders and patterns through base stacking. Each base pair is connected through a hydrogen bond. A and T use 2 hydrogen bonds and C and G use 3. These bonds are weak. Think about the difference when you hold somebody's hand. It's enough to keep you guys together, but you could always let go, versus being handcuffed to somebody. Holding hands is how hydrogen bonds work. A DNA strand is shown here. Here's an example of deoxyribose connecting to the phosphate group at 5' carbon location. The next nucleotide connects at the 3' carbon. Let's discuss major and minor grooves. The major grooves are where the backbones are far apart, and the minor grooves are where they are close together. The more space there is, the easier it is for proteins to bind. As mentioned before, DNA is helical, or in other words known as a spiral shape. Looking at these noodles will help us determine left and right-handedness. As you can see, this rotini noodle is right-handed as the spiral goes up and to the right. This cavatappi noodle goes up and to the left, so it is left-handed. So due to the negative charge of phosphate group, DNA has a negative charge. If DNA was placed in a concentrated saline sodium solution, it would begin to become more stabilized due to the positive charge of the sodium. The higher stability of the DNA allows for an increase in temperature before denaturing occurs, which we can see in this graph. The green line represents the normal, and then the red line represents the higher salt concentration. Here are my references. Thank you.